Hello everyone, my name is Hassan and for the YSP BlueSciCon, I'll be representing the comparison of biotic, abiotic and technological signals based on their statistical complexity and entropy. And for the YSP, my mentors were Dr. Stuart Bartlett, Dr. Anderson Cleaves and Dr. Anirudh Prabhu, and thank you to all of them. So the main problem we were focusing on for our YSP project was how do we approach a signal that is unknown to us? How do we know that it is a sign of life or it is a sign of something else? And I think that this is really important for humanity as we seek to find extraterrestrial or alien life beyond Earth. So this is the process we followed to do our project. So we started off by collecting data, and this included different kind of data. It might be biological signals, abiotic signals, or technological, cultural in nature. And so then we moved on to pre-process this data so that it was better prepared for further analysis. We truncated it, we normalized it, and we applied for a transform rate. And then we got to the main part of our project, that was to calculate different metrics like entropy, complexity, and so on. And then we changed the time series to better reflect the condition in which we will likely receive these signals, and then we made comparisons with within the time series. So we collected very different kind of data that included animal sounds, data from breath to listen, atmospheric data like temperature, dew point, and etc. We collected human speech in different languages. We collected music and songs and economic data. So before we pre-processed our time series, this is how our time series looked. You can see there are a lot of oscillations and a lot of data points. And this is the window where we manually calculate to pre-process the time series, and after resampling it and choosing correct parameters and truncating the time series, we get this. And you can see that the data points have reduced to just 14,000. And then we discretize this time series, and this is how it looks after that. So then we moved on to perform the fast Fourier transform on our data, and to get the characteristic frequency and its magnitude for a particular time series. So we calculated all sort of metrics to better understand our time series, and this included basic statistical parameters like mean, median, curtices, and things like that. And we calculated different kind of entropies, and we also calculated the statistical complexity, which is the main part of my presentation, and we also calculated zip compressibility and so on. So after analyzing these time series, we found that these time series clustered really well into different parts of this graph. For example, here we can see that the living time series for the biological time series have low entropy and low complexity, and for technological signals we have a high complexity and low to medium entropy, and for non-living or abiotic signals we have high complexity and high entropy. So our work can certainly improve in the future by introducing more data, and it will help us get more accurate results and give us deeper insights into the analysis. And we can introduce a wider range of metrics, different methods from complexity and from information theory. And we can also try to use a wider range of the kernel with parameter k for calculating statistical complexity. And we can also use different signal processing methods, for example, the Wiener distribution function and things like that. We can also introduce machine learning techniques, which will help us get better accuracy with our analysis. Thank you all for tuning in.